biological systems are a labyrinth of chemical reactions occurring simultaneously at all times. Some chemical reactions break down complex organic compounds to recycle nutrients, others construct macromolecules from simpler reactants. All life forms have enzymes. Enzymes are specific proteins with the mission of speeding up chemical reactions by lowering their activation energy. Within each enzyme is an active site where specific substrates bind to. These active sites allow the chemical reactions to be catalyzed using a variety of methods. The presence of enzymes allows chemical reactions to proceed at a desirable pace so that the demands for nutrients of a biological system can be met. As enzymes are important vehicles driving chemical reactions, it is crucial for scientists to understand the mechanism of their functions in order to predict their rates of catalysis. For simplicity, it is assumed that an enzyme-catalyzed chemical reaction can be accurately represented by the following equation as displayed on the screen, where E is the amount of free enzyme, S is the substrate concentration, ES is the substrate-enzyme complex formed as an intermediate of the enzyme-catalyzed reaction, P is the product, K1 is the rate constant of the forward reaction forming ES, K-1 is the rate constant of that reverse reaction, and K2 is the forward reaction making the products from the enzyme-substrate complex. For mathematical derivations, the following equations are obtained, where Vrx is the chemical reaction rate and E sub naught is the initial enzyme concentration. As we take the limit of S minus K approaching S, we see the Vrx approaches its maximum, and therefore this is defined as Vmax. Substituting Vmax into the previous equations, we obtain this new equation as displayed at the bottom of the screen. This graph is the result of the mathematical equations we have described earlier, and as you can see, Km is the concentration of substrate at which 1/2 of the velocity at maximum is seen. In this experiment, the purpose is to characterize enzyme catalase chemical kinetics at different enzyme concentrations. Enzyme catalase is a common enzyme found in almost all biological species because it decomposes the toxic H2O2 into water and oxygen gas. In order to characterize catalase chemical kinetics, we will attempt to calculate its Vmax and Km. As catalase concentration increases, Le Chatelier's principle predicts that more enzyme substrate complexes will be produced, thus increasing Vmax and Km. Therefore, if the concentration of catalase increases in catalase-catalyzed chemical reaction, then the maximum theoretical rate of reaction and the value of Km for that reaction will subsequently increase. In this experiment, enzyme catalase from bovine was used. We attempted different dilutions, 1 to 100, 1 to 10, and 1 to 5 to see which concentration worked best. 3% hydrogen peroxide was used as the substrate. Different label plastic cups were used as the chemical reaction sites for enzyme catalase. In each cup, we filled up first with 10 milliliters of hydrogen peroxide, and then as soon as we put in 1 milliliter of the enzyme catalase, we started timing the reaction. After a desirable amount of time, we put in 10 milliliters of 1 mole of sulfuric acid to denature the proteins and stop the chemical reaction. After a chemical reaction was stopped, a 5 milliliter sample from that mixture was extracted to a titrating cup, at which point titration took place. Titration was done using magnesium permanganate. Magnesium permanganate reacts with hydrogen peroxide to form a clear substance. Therefore, when the titration mixture turns purple, that means that all the hydrogen peroxide has run out, and that's the end point, telling us how much magnesium permanganate is used, correlating to how much of the hydrogen peroxide was left after the reaction took place with enzyme catalase. The hope was to use the same method for different times and different concentrations of enzymes in order to graph the reaction over time and tell what the kinetics of catalase is. Using the procedure just described, no sufficient data was obtained to perform a calculation due to experimental restrictions. For some unknown reason, the H2O2 did not decompose upon exposure to catalase. At least 3 trials were conducted to measure the concentration of H2O2 prior to catalase exposure and after catalase denaturation. The results showed that the concentration remained unchanged, thus inferring that no decomposition occurred. Because of that, we took the liberty of creating representative data ourselves. Shown on the screen is what a graph of H2O2 decomposition over time should look like had we gotten any data. Then using linear interpolation, we can calculate the slope at each data point and then plot that into a graph of rate of chemical reaction, which is the slope, versus the concentration of the substrate, which is H2O2. Using this newly created graph, we can approximate Vmax. Then, using the value of half of Vmax, we can project that y value onto the curve and find the corresponding x value, which corresponds to the value of the H2O2 concentration that represents Km. Using the method just described, we have found both Vmax and Km, the 2 quantities necessary to characterize any chemical reaction that is catalyzed by an enzyme. Now let's take a look at the enzyme dependence of Km. Following the prediction of hypothesis, as the enzyme concentration increases, so does Km, and that is what the graph on the screen is showing. Now let's take a look at the possible experimental errors that prevented the lab from working. First, the catalase used had been stored for too long. Second, the catalase used was at a temperature that was too cold to allow a quick chemical reaction to be observed. Third, the concentrations of the enzymes used were too diluted to observe any measurable effects. And finally, the experimental apparatus might not have been sensitive enough to detect small changes in volume that would have been necessary for the measurement of H2O2 decomposition. In conclusion, the hypothesis was neither supported nor refuted because no measurements of H2O2 decomposition over time were obtained. More sensitive experimental apparatus and new chemicals should be used in the future to complete this experiment for a higher likelihood of obtaining results.